University of Sabah, the central dogma of genetics. The central dogma of genetics is an explanation of the flow of genetic information within the biological system. So it was first stated by Francis Crick in 1958 and is restated in a Nature paper published in 1970. First up, transcription. Transcription is the process by which the information contained in a section of DNA is transferred into a newly assembled piece of messenger RNA. Translation. Eventually, this mature mRNA from transcription finds its way to a ribosome where it is translated into a protein. Finally, DNA replication. So this is the process where a DNA is duplicated and makes an exact copy of itself. And here for a special report about DNA replication, it's now welcome our geneticist / molecular biologist / professor / medical doctor, Denzel Zalvera.
Thank you, Romeo. DNA replication. DNA replication must occur in order to faithfully transmit genetic materials to any progeny of any cell or organism. Replication is carried out by copies of group of proteins called ribosome, which consists of helicase that unwinds the superplex as well as the double-stranded DNA helicase. As we can see here in this diagram, this double-stranded DNA is being unwound. This helicase is unwound and then replaced by the polymerase. DNA polymerase. This is as is. In DNA polymerase, then associated proteins will insert, just like what I said, DNA helicase goes out, DNA polymerase insert. Insert a new sequence of nucleic in a specific manner. This typical process takes place during the S phase of the cell cycle. Thank you very much. Back to you, Romeo.
Thank you very much, our resident geneticist / molecular biologist / professor / medical doctor, Denzel Zalvera. And now let's head on to the next report about translation. We have a very, very special interview here. We're really glad that he actually made an effort, arduous, to be interviewed. Let's now welcome Doctor from the Nowhere Neverland Medical Center. I don't know where that is, but it's nowhere. So let's head on to a special report about translation. Good morning, everyone. Thank you very much for this special report. So I'm going to editor MD. Today I'll be discussing with you the process of translation, which is the conversion of mRNA, into amino acid or protein. So mRNA, which is from the process of transcription, will find its way to a ribosome where it will be translated. So the site of translation will be in a ribosome. So guys, just imagine that this is a ribosome because I don't know what a ribosome looks like. So in prokaryotic cells, however, prokaryotic cells, bacteria, which have no nuclear compartment, the process of transcription and translation are getting together. But let us not focus on that. Let us focus on the eukaryotic cell translation. So in eukaryotic cells, the site of transcription, which is in the nucleus, is usually separated from the site of translation because the ribosome is located in the cytoplasm, either free or in the endoplasmic reticulum, either in the rough endoplasmic reticulum. So, so the mRNA must be transported out of the nucleus into the cytoplasm, which it can be bound by ribosomes. The mRNA is read by the ribosome as triplet codons, triplet codons, and usually beginning with an AUG. You know AUG? AUG codon. for the start codon or the initiator methionine codon downstream of the ribosomal enzyme. So complexes of initiation factors and elongation factors make amino acylated transfer RNAs or that is actually the tRNAs into the ribosomal mRNA complex matching the codon in the A to the anticodon on the tRNA. So thereby adding the correct amino acid in the sequence in G. As the amino acids are linked into the growing peptide chain, they begin folding to the correct conformation. Translation ends with UAA, so the UAA. This is actually the stop codon, which is the endpoint of the translation. So UGA or UAG. UAG. Take note of these three codons. These are the stop codons. This is the start codon. Okay? So the nascent polypeptide chain is then released from the ribosome as a mature protein. So from the ribosome, the protein will go up. It will be released as a mature product. So it's already utilized by different parts of our body and different parts of our cell. And from then on, there's nothing to worry about. Okay? So let's have a review first. So this is the mRNA, which is freshly made from the transcription process, and it will actually find its way from the nucleus to the ribosome, which will be inserted in the ribosome, and the start codon, which is AUG, will initiate the translation process and will start converting the mRNA into an amino acid sequence. And as soon as the translation is finished, either the three stop codons will stop the translation process and will eventually produce a mature protein, which will be released into the cytoplasm or anywhere it will be released. So that's my report. Translation, you remember translation, which is the conversion of mRNA into protein. Now back to you, Romeo. Thank you very much for that wonderful report, Dr. George, from the Lower Neverland Medical Center. I still don't know where that is. So let's now head on to our field report. That's outside the University of Sapacamoo. For our field reporter, Ms. Shirley Skerlan. Paso!
Thank you, Romeo. We are now live at the University of Sapacamoo with Dr. George III, who will be talking about transcription. Doctor, can you tell us about transcription?
Sure. Transcription, as you know, is actually the conversion of DNA into the RNA sequence, which happens in the nucleus of the cell. So, after this process, the RNA that has been transcribed would be in turn delivered to the ribosome, ready for translation into a protein. So, let us watch this little video that I made myself to understand more about the process of transcription.
Transcription is the first step of gene expression in which a particular segment of DNA is copied into RNA by the enzyme, RNA polymerase. Both RNA and DNA are nucleic acids, which use base pairs of nucleotides as a complementary language that can be converted back and forth from DNA to RNA by the action of the correct enzymes. So during transcription, the DNA sequence is read by an RNA polymerase, which produces a complementary anti-parallel RNA strand as opposed to DNA replication. Transcription results in an RNA complement that includes uracil. In all instances where thiamine would have occurred in a DNA complement. Also, unlike the DNA replication, where DNA is synthesized, transcription does not involve an RNA primer to initiate RNA synthesis. So transcription proceeds in 5 or 6 steps, each moving like a wave along the DNA. First, one or more signal factors initiate a transcription of a gene by enabling binding of RNA polymerase to a promoter DNA. Next, RNA polymerase moves a transcription bubble like the slider of a zipper, which splits the double helix DNA molecule into two strands of unpaired DNA nucleotides by breaking the hydrogen bonds between complementary DNA nucleotides. Third, RNA polymerase adds matching RNA nucleotides that are paired with complementary DNA nucleotides of one DNA strand. Fourth would be RNA sugar phosphate backbone forms with assistance from RNA polymerase to form an RNA strand. Fifth, hydrogen bonds of the untwisted RNA plus DNA helix break, freeing the newly synthesized RNA strand. And last, if the cell has a nucleus, the RNA is further processed, addition of a 3' UTR poly-A tail and a 5' UTR cap, and exits to the cytoplasm through the nuclear pore complex.
Thank you very much for that beautiful report. We share Luis a lot. Now, there are actually some cycles in the central dogma, which is like access a special pathway, for example, the conversion of RNA back into a DNA template or a cDNA. That process is actually called a reverse transcription. Now, to know more about the reverse transcription process, let's watch this simple video of the reverse transcription polymerase chain reaction. So this is me again, live from the University of Sapamango. Romeo Shapiro, your news anchor for today. Thank you very much. Have a good day.
Oops, and